Good afternoon and welcome everyone to BioAlberta's mentorship series. My name is Rob Stoddard and I'm the President and CEO of BioAlberta, and I want to thank you for joining us today. BioAlberta is a member-driven and funded voice for Alberta's life sciences sector. Our mission is to grow a vibrant life sciences sector to diversify the economy by leading Alberta's recovery. We support and connect researchers, students, SMEs, industry leaders, and funders to advocate for our success. Events like today's webinar are part of our mentorship series, which offers access and insight from industry leaders who share their expertise and experience to help grow our sector. Today's webinar is number 4 in a series of sessions by the Metabolomics Innovation Center at the University of Alberta, focusing obviously on metabolomics and life sciences. The workshop topic today is metabolomics in the agriculture and forestry sectors, 2 traditional economic powerhouses for Alberta who have always been on the leading edge of using technology to move their business forward. Before we get too far and get into introductions, I just want to go over a couple housekeeping items. First of all, if you can keep your microphones muted at all times, that would be helpful. It's easier for our hosts and our panelists to keep the distractions down. We do have some questions already submitted. Thank you for those who came in. But if you'd like to ask a question of the panel today after Dr. Wishart's presentation, please type your question into the chat and we will ask your question amongst the others. I expect you already know Dr. Wishart. That's part of the reason why you're here and part of why you've joined our mentorship series. We're going to have the panel introduce themselves for about 30-40 seconds to walk through their bio very quickly. We're going to have Dave Wishart anchor those introductions, and then he's going to launch straight into his presentation. So with that, I will turn it over to the panel.
Hi, everyone. Pleasure to meet you all. Thanks so much for the opportunity to be here. My name is David Lloyd. I'm the CEO and one of the co-founders of a startup company based out of Calgary called FredSense Technologies. We're a water instrumentation company that's commercializing portable field kits to help the water sector at large understand their water quality faster than before, using biology as the main toolkit. We build in vitro and in vivo systems that allow live organisms to respond to different types of compounds that can be found in the water and report that to users electrochemically. We measure things like arsenic. We look at COVID-19 analysis in wastewater. And I personally have a background in biochemistry and synthetic biology, so really excited for the conversation today and hearing the presentation.
Hi, everyone. This is Shiv Prasher, and I'm in the Department of Bioresource Engineering at McGill University in Montreal. And I'm also very pleased to be part of this workshop. And my main area of research is environmental engineering, mostly looking at non-point source pollution from rural areas. And when I say non-point source pollution, I mean both conventional and emerging contaminants. By conventional, I mean pesticides, nitrogen fertilizer, phosphorus fertilizer, and so on. And by emerging contaminants, I mean antibiotics, hormones, and stuff like that. And I also look at best management practices. What can we do to reduce this pollution from getting into our water resources and contaminating them? So in short, that's basically what I do. Anything to do with water contamination and taking measures to address that situation with the help of farmers and other stakeholders. So I'm very happy to be here today. Thank you.
Thanks very much, Shiv. Thanks, David, as well. I'm Dave Wishart. I'll be giving you about a half-hour presentation today about metabolomics and the environment. As Rob was suggesting, this is the 4th of our series. So last week we did one on agriculture. The week before that was on precision medicine, and the week before that was just a general introduction to metabolomics. Some of you may have been at all 4 of these meetings. Most of you, I assume, haven't, and so I'll be spending a little bit of time introducing you to the topics for today. My own background, I'm a professor in both biological sciences and computing science at the University of Alberta. I'm one of the co-directors of the Metabolomics Innovation Center, and I'll refer to that as TMIC, which is the abbreviation for the Metabolomics Innovation Center. So without further ado, I'm looking forward to the meeting today and interacting and answering questions and working with our team today. I'm going to switch on to share the screen, and I'll just do sort of a video and sound check. So can everyone see those slides? Good. And I'll shift to the full screen, and I'll use, I guess, the next half hour or so to tell you a little bit about how you can use metabolomics in understanding, assessing, testing, evaluating the environment. And when we say the environment, that can work in both forests, fisheries, aquatic systems, air, soil, anything that we stand on, drink from, breathe. And as you say, it's a fairly broad topic and obviously impacts fish and wildlife, macrobiotic systems and microbiotic systems. So it's a broad topic, and it's a lot to cover, so I'll get right into it. So those of you who are new to this workshop or new to the field of metabolomics, this is just a quick definition of it. Metabolomics is the study of small molecules, or metabolites as we tend to call them, and anything that we can find in a biological sample. So that can include tissues, it can include stuff that we find in animals, it could include their excreta, it could include plant tissues, soil, it can also include water and air. What metabolomics offers people is a very rapid, inexpensive, and very comprehensive way of quantitatively characterizing chemicals. So that could include things like primary metabolites, things like amino acids or sugars. It could include secondary metabolites. Many phytochemicals are considered secondary metabolites. It can also include synthetic chemicals. That might be herbicides or pesticides. It could include drugs. These are things that end up in our bodies or the bodies of animals or insects or plants. It can include pollutants or contaminants. These might be things that come from burning. It might come from spills. It might come from detergents or cosmetics or dyes that we use to clean our clothes or color our hair or whatever. So these are all of the chemicals that typically can be seen, quantified, or measured through metabolomics. One way of thinking about metabolomics and its connection to other omics fields is this pyramid of life picture, where at the base of the pyramid we have the genome, and to study the genome we use genomics. DNA codes for proteins, so proteomics is the way that we study proteins. And proteins synthesize or break down metabolites. And so at the top of the pyramid is the metabolome, and the process of studying those metabolites is called metabolomics. As you climb up the pyramid, what you'll find is that there's a different degree of influence. What you eat or breathe affects your metabolome. What is in the environment affects the metabolome. And so in that regard, the metabolome is a very sensitive indicator of environmental contamination, environmental interactions. The analogy is hopefully what you eat or drink will not affect your genome, so it's a very stable thing, whereas the metabolome changes very rapidly with time or exposures. Same sort of thing is that physiology is also heavily influenced as you go up the pyramid. The genes are essentially identical in plant or animal cells, regardless of the tissue. But with different tissues, you'll find very different metabolites or metabolomes. So the metabolome of a leaf is very different than the metabolome of a root, which is very different than the metabolome of a flower. You'll also see very different metabolomes for different types of organisms between plants and insects and animals. What's appealing to a lot of people about metabolomics is it lies at the interface between the environment and the genome. And so in that regard, it gives you a really useful quantitative readout of the phenotype. And that's what I sort of illustrated in those silhouettes at the top, that the metabolome sits at the interface between the environment and the genome. Now, to do metabolomics, there's a variety of technologies that we have to use. A lot of them are borrowed from the field of analytical chemistry. So we do a lot of separations with liquid chromatography, high performance or ultra high performance liquid chromatography, capillary electrophoresis. There's lots of microfluidic techniques that are used. There's also a lot of instrumentation like mass spectrometers. So liquid chromatography mass spec, triple quad mass spec, QTOF mass spec, Fourier transform mass spec, inductively coupled plasma mass spec, all of those abbreviations for words that probably mean nothing to most of us, but these are the tools that we regularly use in a metabolomics lab. We also use NMR spectroscopy, gas chromatography, or GC-MS, and even infrared and ultraviolet spectroscopy are employed. So it's quite an array of instrumentation. And to get all of that instrumentation in one or several places is expensive. This is what the Metabolomics Innovation Center does, is it's brought all of that instrumentation into several facilities or nodes located across Canada. Originally, TMIC started in Edmonton and drew in other nodes from Victoria, and then others from Calgary to Montreal to Hamilton. Its mandate is to provide metabolomics services and perform cutting-edge science and advanced technology support. It is now called Canada's National Metabolomics Lab. It is supported by the Canada Foundation for Innovation as well as Genome Canada. And the website is given in red here. It is nationally distributed. There are 9 different nodes for TMIC. Most of the nodes are located in Edmonton at the University of Alberta. There's the administrative node. I have a node, James Haranek, Liang Li, Michael Overdwin, all at the University of Alberta. Ian Lewis is in Calgary, at the University of Calgary. David Goodlett, the University of Victoria. Christoph Borchers is at McGill University, and Philip Ritz McKibben is at McMaster. So we cover a fair number of provinces, and that's why we have sort of a national mandate. So in terms of TMIC and environment and metabolomics, there's lots of applications that our group and node members have done, and I'm going to give you a few examples. And it's not just what you can do with TMIC, it's what you can do with metabolomics in general. Metabolomics has been used in assessing forestry health, forestry pest interactions, assessing water quality and contamination. It's been used in wildlife monitoring. Long history in Alberta of using metabolomic methods to analyze oil residue and oil sands products, working in bioproduct development from anything from agriculture to processing waste materials. A field of science called exposomics or exposures, which is more the interface between environment and human interactions. Analysis of herbicides and pesticides, heavy metals, microorganism and microbiome profiling as it relates to soil or water, and then looking at the bacteria through chemical analysis, looking at gene-environment interactions, which, as I said, is the real strength of metabolomics, looking at also volatile compounds as well. So I'll give you, I think, 5 or 6 examples over the next 20 minutes here, and trying to cover some applications in environmental studies. I won't be able to cover all the ones that we've ever done, but just some examples, and hopefully that will invite some questions or queries. So one was in the area of, if you want, wildlife metabolomics, and in particular, we are interested in chronic wasting disease. This is a disease that's affected deer, elk, large number of cervids and cervid populations, and trying to understand what happens and if there's a way of detecting chronic wasting disease early. Typically, to go from a healthy animal to a very sick or dead animal is about 18 months. And our interest is to see if there's ways of either looking at blood samples or deer droppings, elk droppings, that would allow us to detect infected animals long before they show any symptoms. And if you can detect before symptoms manifest, then that potentially reduces the spread in, say, elk herds or between animal to animal during the rutting season. What we found is that with metabolomics, we could detect fairly significant changes between the animals that were healthy, that's in red, and the animals that were sick and at different time points during that progression. In particular, this is just showing how these things change over time. So we started with healthy animals, inoculated them, and then followed how their metabolites, looking at their blood levels, changed. And within 7 months, there was a striking change, which suggested that we could determine through a simple blood analysis what animals were infected. And then from 7 months to 12 months into the fatal stage at around 18 months, yes, there were changes, but in fact, they were more minor. And that again suggested that metabolomics might be a more powerful approach to detect animals that were infected. Now, obviously getting blood from wild animals is difficult, unless, of course, maybe you're hunting them and then you're able to take blood samples. But what is generally easier if you're trying to do environmental sampling is to look for detritus, that is deer, elk droppings. And so we started looking at droppings from animals, in many cases, these were elk farms or other captive animal herds, where we could look at droppings where we knew the animals were infected or knew the animals were not infected. What we're showing here is essentially a metabolomic analysis of elk droppings, and we're looking at the levels of different metabolites that distinguish between positive and negative animals. And this is part of a CWD project that was led by Dr. McKenzie at the University of Alberta, Deborah McKenzie, who's with Biological Sciences. One of the striking things that we found was that one particular compound, butyric acid, was significantly reduced in animals that had chronic wasting disease. And this suggested that it might even be possible to develop a very simple one compound test to distinguish between animals, or at least areas that have deer and or elk or other cervids that have CWD in the population. We looked at other combinations where we tried butyric acid, amino acid leucine, another carnitine, decanol carnitine, and xanthocene. The performance for that test was around 91% correct, which is very good. But interestingly, just using the single organic acid, butyric acid alone, was about 83% correct. So this suggests that, in fact, we may have a very simple, useful test which would allow field analysis to detect animals that are positive for chronic wasting disease. So that's a quick example of using metabolomics to identify, in this case, infected animals that allows potential interventions to take place, or a monitoring of the situation. Now, you can also use metabolomics to look at plants, in this case, plant-pest interactions. We have a couple of examples here. So one is to look at canola, which is obviously a big crop in Alberta. And this is where we're looking at a technique called gas chromatography or tandem gas chromatography, GCGC analysis. And we're trying to detect Bertha armyworm. This is a caterpillar or moth that can infect canola plants and can damage them quite severely. And they spread quite quickly. So what we're looking at are volatile. These are compounds that evaporate quickly, that are produced by plants, and any of the scents and aromas that you smell in flowers or other plants, these are the volatiles that your nose detects. And this is essentially showing all the types of volatile, literally hundreds of different alcohols, aldehydes, short-chain fatty acids, alkanes, terpenoids that are all produced from canola, some of which tend to attract. What we found is that there are certain signatures that are associated with canola plants, some of which are clubroot resistant, some of which are clubroot susceptible, but which attract or were produced by certain strains of canola which strongly attract Bertha armyworms, and then others which basically are indifferent. So this suggests a way of not only selecting for canola species or varieties that are essentially more resistant or less likely to attract Bertha armyworm, the moth, or the caterpillar. And this, again, offers, I think, a hope or a way of evaluating canola selection strategies. Now, in addition to agricultural crops, there's also studies that we can look in terms of forestry. One that we're obviously dealing with in Alberta is the mountain pine beetle. This is an infestation that started in British Columbia and has migrated across the mountains and into Alberta over the last 5 or 10 years. And what we're interested in is trying to identify the trees that seem to be resistant to mountain pine beetles. You can occasionally find a few trees that are nice and green, surrounded by a forest of the brown dead trees. And it appears that this has a lot to do with the odorant molecules, the volatiles that are produced by pine trees. And by looking at the terpenoid compounds that came out and that were measured through metabolomics, we found that certain compounds, namely limonene, beta-phalandrine, and gamma-terpenine, had essentially a strong repellent effect that prevented mountain pine beetles from either landing on the tree or even attacking the tree. Particularly limonene and phalandrine were the best ones, and these are ranked #1. So again, this suggests that either by breeding trees or selecting trees or using a combination of different fertilizers or nutrients, it's possible to create trees that are essentially resistant to mountain pine beetle. Now, a similar situation is also going on with spruce budworm and these worms attacking spruce trees, which are also part of the Alberta landscape. And once again, there seems to be a distinct preference for certain types of compounds, namely what are called terpenes or terpenoids. These are the things that produce, say, the pine scent that you'll find in certain kinds of deodorants, or obviously if you walk through a pine forest. But in this case, alpha-pinene, one of the main terpenoids that gives you the pine smell, actually can be quite useful for preventing spruce budworm attacks. There are certain classes of trees and spruce families that we found that had very distinct clusters and odorant profiles. And what's more, when we compared the terpenoids with the polyphenols, we found that the terpenoids were much better at preventing spruce budworm attacks than the polyphenols. And again, this is an important discovery, if you want, because for a long time people felt that the polyphenols were critical for battling against spruce budworm, but in fact it has more to do with the scents and with certain compounds like alpha-pinene. In other words, it's sort of like the spruce, by producing pinene, disguises itself, and it makes the spruce budworm think that these are pines and not spruces, and so it avoids them. A third example is to look at the metabolomics or exposure and exposomics of river water. So we've looked at plants, we've looked at forests, we've looked at animals, but obviously all of them use water, all of them need water, and so do we. So at TMIC, we've been working for a long time at developing high quality comprehensive analyses of pesticides and also of herbicides. Right now, we're able to measure about 60 of these different molecules, and we're slowly ramping up to measure close to 100. These are quantitative analyses, and so we can do pesticide residue analysis of plants, of soil, of water. And given that the watershed captures a lot of these things, and many of these compounds are stable for a long time, that sort of analysis is obviously quite useful. We've also looked at other molecules, in this case looking at the North Saskatchewan River, which is the river that flows through Edmonton. And we did what's called an untargeted analysis through using liquid chromatography mass spec, and to identify a variety of compounds. Some of the things that we found are large numbers of polyethylene glycol, or PEG, surfactants. These are things that are typically added to paints. They're found on clothing. They're also used in oil well work. They're pretty ubiquitous, but what was surprising was just to see how many different PEG varieties were in our water. Other types of surfactants, plasticizers like BPA also are in our river. We have flame retardants, hair product compounds, bactericidal compounds. We also found a lot of compounds that are related to the synthesis of fentanyl and opioid drugs. So evidently there are a few places that are dumping compounds into the river in the North Saskatchewan illegally. We can look at seasonal variation of these contaminants, and we looked at this over several months and looking at how things changed both from June to September. And we looked through the essentially water clarification purification process that was happening at the Devon water treatment plant, and found very distinct differences both in terms of what accumulated and where it accumulated and at what time of year it accumulated. Certainly the wastewater was generally more infected with more material, but certainly the river water was also more infected at the beginning of June than it was in September. We can use not only liquid chromatography mass spec to look at essentially non-volatile compounds, we can also look at volatile compounds like hydrocarbons, things that evaporate, things that give you the oil smell or the gasoline smell. In this case, we're doing GCGCMS analysis of river water shortly after rainfalls, and to look at storm drains as they come in. So we're collecting samples not directly from the river, but at the storm drains. And what we found is a lot of hydrocarbons, many of them from fossil fuels that we get from diesel and gasoline, but also from asphalt and tar, as well as degraded rubber and plastics. And they certainly tend to spike up after a rainfall, and these are the things that are deposited on your roads. So this is all concentrated and washed into the storm drains. It's also an issue with regard to plastic and what sort of containers should be used to collect samples from water studies. This is just illustrating that if you put your water samples into a general plastic tube, you will get contamination. And this is just how sensitive the systems are, where we can get essentially leaching of plastic into distilled water that we got from the lab. So if we're going to do environmental analysis, and as most people doing environmental analysis know, you really need to either collect the samples in glass or Teflon containers, just so that you aren't producing essentially artificial contamination. So from river water, we can go to ponds and mining sites. Of course, many of you are aware of the tailings ponds in northern Alberta. And this is an analysis that was done on some of the tailings ponds looking for naphthenic acids with the water that's been processed. And these are found in not only the oil sands and the tailing ponds, but they're also just found in bitumen. So these naphthenic acids are important because they are known to cause toxicities in aquatic systems. And this is just illustrating what's happening with conventional mining, the types of carbon systems and naphthenic acids that we found, how big they were, where they were found, versus the SAGD drilling, which is not really a mining process, but it does produce material that can eventually end up in the water system. Now, we've already talked a little bit about pest-forest interactions, but there's also examples of pure forestry metabolomics. In one of the studies that we did, we were interested in just looking at how trees respond to drought and other types of stresses. In this case, we developed assays to measure a large variety, more than 150 different compounds in leaves or needles from spruces, also from other parts of the plant. We could look at amino acids, organic acids, sugars, polyphenols, known plant hormones, terpenes, terpenoids, and lipids. We were looking in both spruce and pine, and this is just sort of illustrating the difference between how pine responds to at the bottom and spruce responds to drought. So in the case of spruce trees, glyceric acid and serine are decreased during drought. In the case of pine, beta-hydroxybutyrate and tryptophan increase significantly. In the case of tryptophan, it looks like it's actually used as a source molecule for certain hormones to help it respond to drought. And generally, it seems that pine trees respond to drought better than spruce trees. Now, we've been collecting metabolomic data for years through TMIC, and we also have to interpret the metabolomic data and work with clients and collaborators to give a better chemical or environmental understanding to what we've measured. And so as part of that effort, we've been building databases for environmental metabolomics. And part of this has to do with the fact that there's obviously lots of compounds that we produce synthetically, naturally. We need to track those, and we have very large databases of those. When those compounds are released into the environment, deliberately or accidentally, they get transformed. If we eat them, inhale them, or ingest them, if our microflora ingest them, if the soil ingests them, or if sun and solar radiation or oxygen or other things get to them, they get transformed. And it's those transformation products that produces the sort of the green goo that I've illustrated, but it's very diverse. And so while there might be 300,000 or 400,000 well-known compounds, we estimate there's around 5 million less known or mostly unknown compounds that potentially can be detected in the environment. As part of that effort, we've been collecting information on exposure chemicals, things that are generally more abundant, tracking this in a database with our colleagues in France at the Institute or the International Agency for Research on Cancer in Lyon. Together with them, we've created an exposome database looking at pollutant biomarkers, their concentrations in blood and urine, and references along with detailed information on their structure and chemical properties. We've also been building a database on contaminants. Right now there's about 55,000 contaminants, and we're adding to it every month. We have lots of detailed information about these molecules, what they look like, information about their spectra so that we can identify them more precisely. A lot of the compounds that are in this database are in no other database. And that's made it particularly challenging because we often have to draw all the structures by hand in order to fill out this particular database. So that kind of wraps up some of the examples of where we're using metabolomics or how we've used metabolomics to help understand the environment, to help understand water, forest, pest interactions, wildlife. We've had other applications in soil. We've had applications in pollution. And it's not only the analytical services that we provide, it's also the databases and the data interpretation. So as a national facility, we can offer people expert advice on using metabolomics for environmental analysis and contaminant testing. We are ISO or almost ISO certified by the end of next month. We can do a broad array of analytical and chemistry services. We have large collections of software and databases. And we're also developing tools and techniques that allow us to take the tests into the field, so lab to site translation skills. We've worked with many groups, and in many cases we've done complementary or free small-scale pilot studies to help get initial data sets, to help explore potentially either business ideas or grant application ideas. We very much want to work with people. We collaborate with dozens of people around Canada and around the world every year. Joining us today actually is Jen Reed. She's our business manager, operations manager for TMIC, and has been with us for many years. So we certainly invite you to contact Jen. Her email is given here. And then we can also field queries through the website, which is metabolomicscenter.ca, which is given below. Also just want to thank my colleagues. These are the node leaders and co-directors at the Metabolomics Innovation Center: Liang Li, Christoph Borchers, Phil Ritz-McKibben, Michael Overdwin, James Hernick, Dave Goodlett, and Ian Lewis. And then also our funders from Genome Canada, the Canada Foundation for Innovation, NIH, CIHR, Western Economic Diversification, and NSERC. So thank you very much.
David, thanks for that. That's a very informative overview, and really appreciate the level of information. Want to remind everybody to take a look at the chat box. There's lots of rooms for questions in there now that David's finished his presentation. Feel free to put your questions in the chat so we can ask them to the panel. While you're doing that, let me get things started off with a general question about about government's reaction or government's acceptance of these new tools and new studies, and how when it comes to the drivers at a regulatory level, is this a case of technology and the capacity of metabolomics to push regulators to look at different tools and accept different methods, or is it more of a pull by regulators and industry researchers responding?
I don't know, Shiv, would you like to try that one?
Sure. I think this is a question that David and we were recently involved in a project, and we kind of faced this situation that if we are coming up with new techniques, because most of the time the sampling is done using conventional methods that have been tried and tested for many years, and according to the regulations, then they're supposed to be following those approaches. These newer approaches, I think they need to be, the regulators have to come on board before these newer approaches can become part of the analysis that we do for water quality, for example. I think this is something that has to be addressed. I think there's a lot of potential, because we were trying to use genomics and metabolomics in this project for water quality, and the questions were raised if the regulators would buy into these new technologies. So I think it's it's a question that has not been answered as far as I'm concerned. I don't know if, David, you want to add something to it?
Yeah, Dave Lloyd, did you want to make comment on this, or?
Sure. I think one thing that we're seeing within the realm of water quality analysis and the regulatory compliance limits, particularly here in North America, is that we're finding more and more a trending down of regulatory limits and a trending down in the need for sensitivity of analysis. We're learning more about the health and aquatic systems implications of various compounds emerging and otherwise that we're finding within our systems. And there's been several examples of areas where the limitation in being able to implement government policy or different types of regulatory structures is the cheap and available capacity to be able to provide water quality information in a timely way. And Shiv, as you mentioned, the traditional approach of utilizing very streamlined, well understood approaches for sampling and traditional analytical methods has allowed us to get to a certain level, but now there's becoming more and more needs for different types of technologies and systems, whether it be field methodologies or analytical lab methods that will really allow us to get to the next step. And some examples of this are, for example, the class of compounds known as PFAS chemicals, these forever chemicals that we're seeing within water systems, where regulatory requirements are becoming more and more likely to be seen across different jurisdictions, and the technologies and standards and methods that are required in order to be able to do this monitoring really may require new types of technology adoption. And that's where metabolomics or next generation methods like biological sensing systems and other types of field kit systems really have a role to play. And it's in that alignment with regulators that we can really see an opening up of new types of ability to keep communities safe, to understand in a cost-effective way for end users how we can understand our water quality in real time. That's going to become more and more important.
So David, let's just stay with that theme for a second. When you're talking about the water quality that people are exposed to in a municipality or a town, whether it's the drinking water or whether it's water in the general environment, there's obviously a need for a quick turnaround. So in that type of analysis, where is industry at bringing those technology and those testing methods and kits to something that's maybe more, and on the health side we would talk about point of care, but more field testing or faster analysis?
Let me jump on to that question. That's really what's driving us at FredSense, is that much of traditional sampling today is being done through analytical labs, that there are many online analyzers that are available, or different types of field kits that are available for some traditional compounds. But for those that are very low concentration, or are emerging, or are difficult chemically or through traditional types of methods to be able to monitor, this can be very difficult to do in the field. And for many applications, the speed at which we're able to generate these data sets, that we can get this water quality information, directly impacts the usability of the data. And so we see an opportunity for technologies like metabolomics and other types of biological sensing tools to really provide that transition from what may be available today from that analytical lab environment into the field. So utilizing systems that allow you to generate that information right away. Now, there's a barrier to that, in that it needs to be rapid, it needs to be simple to use, and it needs to provide that high quality, accurate data. And that's really the transition that we're seeing more and more driven into industry, is acquiring that quickly and effectively as new technology tools allow us to do that. and then move that technology onto online real-time systems where the challenges become not so much the data acquisition, but the data handling, the usage in databases, like you talked about, David, in your presentation, is gathering the data and turning it into usable insights. So we're seeing that transition more and more, in an opening in industry to accept more of these tools in partnership with end users and regulators alike.
If I can also add to what was just said, that this is also something that we found when we were writing our project, that most of the conservation authorities and people responsible for river health, and so, they wanted something that can be quick, like not waiting for weeks to know what was the water quality, and then valuable time would have been lost. So something, and we did find out from David's group that there are possibilities of field kits that can be developed so the turnaround time could be very short, and so timely actions could be taken for knowing what kind of contaminants are causing water quality concerns in water courses. So I think that's a very important thing. One thing that I also wanted to mention was, and metabolomics can help. Whenever we deal with contaminants, like conventional contaminants, we are looking for very specific contaminants. And they could be contaminants that we actually are not aware of. I'm talking about emerging contaminants and antibiotics getting into the water supplies and other chemicals. And also may not know what their impact could be together on things that that are living in water courses and so on. So metabolomics could actually even open those kind of doors, like what could be a combined effect of inorganic and organic, conventional and emerging contaminants together on the river health. So that could be another opportunity that I see could be very useful.
Shiv, thanks for that, and thanks, David, for that answer. Lots of Daves on the call today. So our first question from the floor is coming from Dave Arndt. So thanks in advance, Dave, for putting this question to Dr. Wishart and to Dr. Prasher. Are there specific or persistent challenges when trying to analyze certain sample types, for example, soil compared to water or other samples?
Yeah, I guess I could maybe answer that one. We call them matrix effects, and it has to do with a lot in terms of the complexity, texture, density, and structure of the sample. So soil is difficult to analyze. Water is easy to analyze. As a general rule, liquids are the easiest to analyze through analytical chemistry. When we look at soil, because of essentially the large collection of humic acids. These are polyphenolic structures that grab onto just about anything and everything. It's hard to pull out certain chemicals. They're just like sponges that hold on to things. So it is a challenge to extract certain types of compounds from soils and humic acids. Obviously, they're solid. So again, we do lots of solvent extractions to pull things out. There's a similar issue with analyzing plants. The tissues, so leaves are soft, petals are soft, seeds are a little harder, bark, xylem, phloem, tissues, roots, those are also a little more difficult. Analyzing microbial samples growing in culture, that's generally easier, but looking at microbes, say, growing in soil, that's a little harder. So there are different challenges, and we often have to come up with a variety of extraction techniques. Each sample is unique at some level, and this is one of the, I think, the strengths in the Metabolomics Innovation Center. Lots of expertise in working with many, many different matrices and many different sample types.
Yep. I think, David, you have touched upon all the good stuff, and I just wanted to add also that in addition to looking at the microorganisms, if we can look at also, for example, other living things like earthworms in the soil, which have to deal with contaminants, because we were involved in a project with, where the sewage sludge was used, and we wanted to use metabolomics to understand, the toxic environment, if there could be, when we use sludge, in the agricultural fields. And so we were looking at earthworms, and then extracting the earthworms, and then looking at the metabolites that could be present where the sludge was applied versus where the sludge was not applied. So metabolomics can also be very useful technique in that sense, looking at another level, not the micro level, but at a macro level.
And I think this is a really good point, whether it's sentinel species or apex species, that either those that are first exposed or those that sort of concentrate the metabolite or compound of interest. But those are often a great way of getting, and at some level even concentrating the compounds of interest from the samples that would otherwise be too hard to extract by conventional means.
Exactly.
Okay. General question. So anybody, any or all of you jump in on this one. Beyond research and discovery, what considerations should be made when moving towards more of a translation of metabolomics-enabled discoveries in the environmental sector? Don't be shy.
Well, maybe I could try and answer that, and maybe inspire a few more comments from David and Shiv. So I think one of the key points that we've heard in terms of translational needs is to have field test ready systems. And I think, as David pointed out, there's a limit. You can't replace $1 million instrument with a $10 instrument. They just don't have the sensitivity that $1 million instruments do have. But if you can, it is nice to be able, at least to facilitate translation, is to make these things into handheld or portable systems. Mm-hmm. We've been working on creating a portable CWD detector, one that would grind up essentially deer droppings or elk droppings, and then allow us to do a quick color test to look for butyric acid. And the only way we could have known that butyric acid was useful was actually doing it on the $1 million instrument, but now you translate it to the $2 instrument. And I think this is historically how a lot of field tests have evolved. We've worked a lot in terms of trying to develop assays that can be portable, experimenting with a lot of chemistry, a lot of biochemistry. And I think David also in FredSense has experimented with and developed a lot of these field ready tests. So maybe, David, you can offer some comments.
Yeah, I agree with all of what you were saying there, David. It's so important and critical in that transition. And it's designing for, what we've really found has been the translation of designing for a very different type of use case than what the analytical lab environment can provide. That oftentimes these are in rugged environments where you may not even have a flat surface to be able to work with, that it requires a very simple to use protocol and procedure. And oftentimes the same solution can be deployed in incredibly different environments. So speaking again back to the point on matrix effects and how different watersheds may even just have different types of compounds. So a lot of our journey at FredSense has been in taking robust systems that work effectively within a lab environment and simplifying them down, creating that common understanding of the components to be able to package them into instruments and hardware that when you go to do arsenic testing or you're doing COVID-19 analysis in wastewater, you have a very simple, ruggedized protocol that works effectively in different types of environments and are simple for operators to be able to implement.
If I can just add to whatever has been said, is I'm also thinking that maybe the regulators or other people have to be involved in these projects so they actually can see how this technology can be helpful, so that it will be more easier for them to accept it, and then for much easier for this technology to become acceptable in regulations or where it's adopted. So I think in addition to what has been said, I think this is also in projects we need to get them involved so they can actually see what they can observe from traditional testing and what they can actually see or get from metabolomics and how it can add value to what they were doing otherwise. So I think that could be another factor that I think will be very useful as we go forward.
Agreed.
So let's build on that a little bit. One of, as part of BioAlberta's sector composition, we have biomass, bioenergy members who are part of our base. And we've talked a lot today about environmental monitoring and environmental testing, but how would metabolomics as a tool improve manufacturing processes in the bioenergy space or bioenergy applications?
I think in many cases, you're trying to look at a few different things. In the course of production, you still produce waste, and so you may want to know what is that's being produced and how dangerous or toxic it might be, what should be done with it. There's also the processes of what have I made and what is in it. So again, this is where metabolomics is really good at it's essentially understanding the chemical composition of something. And also in terms of consistency. So you want to be able to make a product that is identical from day to day, week to week, month to month. It may look identical, it may sort of feel identical, but chemically it could be quite different. And this is something that we see in particularly in food. I see it in the brewing industry, where issues of consistency can vary tremendously. Even though the product might look the same or at a gross level seems the same, chemically it can be profoundly different. It's the same thing even when we just talk about water. Water looks clear, it's probably safe to drink, except that when we analyze it, there's a lot of stuff in there, all of it invisible to our eyes. And so this is what the being able to analyze at a chemical level allows you to distinguish and differentiate things that visually or in a crude sense seem to be identical.
Okay. A question from Ahmed, and if we're getting to the point where if you have one or two more questions, please get them in the chat in the next couple minutes. A question from Ahmed: are there any examples of TMIC current or alumni members' work that made it to markets in the translational space?
Yeah, there's quite a few. We've had a number of people who've been in TMIC who've spun off their own companies, and they're doing actually very well. There's one company called Metasci, where they're selling compounds and mixtures that are being used as analytical standards, reference standards in a wide variety of applications in agriculture, medicine, environmental testing. We have another group that spun off a company to do drug analysis and drug discovery. It's called Omics or OMX. And they've grown to about 45 people, and they're worth millions of dollars. So there's groups that have come from either the technology or from the innovations or discoveries that have been made. We've also helped a lot of other companies move their products out and into systems. So it's a mix. There are people from within TMIC and many people from outside TMIC who I think have done very well.
Shiv or David, anybody notable that comes top of mind in the area of translational research?
Some names, or
I know the challenge. You don't want to start naming names because you're going to forget somebody that's going to hold it over you, aren't they?
Yeah. So Reza Jafari is one who's done things in Metasci. Craig Knox, Mike Wilson in OMX. Neil Taylor with Conomics is another company. And there's Nova MT, which is a spin-off company that Liang Li developed, and he's got several staff, and I can't name all of them involved with that. So I think those are a few examples. Other small startups, Trika Incorporated makes use of, they're working on developing sensor systems that are portable. And that Scott Mackay is one of the alumni from TMIC. So those are just a few names of people that have come from TMIC and have started up successful companies and products.
Excellent. So final question, and this is to all panelists, and then after this question, maybe David, I'll get you just to Dr. Wishart, maybe a couple moments just to wrap up. But this question is for all panelists. So new measurement techniques focus on targeted rapid field tests of a few compounds. Are there other emerging practical applications that involve looking at large sets of compounds, or are these types of analyses primarily performed in research?
If I may, I wanted to comment on this because this is actually very topical for us at FredSense, is that we've primarily focused in the past on developing discrete organisms, discrete biosensor solutions for particular compounds of interest. Often because today what we do in many of these industries is we focus on regulating particular contaminants and then relating that to what's allowable within our systems. But one of the major effects of that, and where many things are going, is not just looking at the chemical makeup, but the health impact of many of these types of compounds. And what does that mixture of these organisms together actually mean through a lot of these tests? And the way that we can do that today is oftentimes through looking at trout tests or other types of biological systems for looking at health data. So using organisms, we're also exploring how could you look at broad classes of different types of compounds and then look at it through a toxicity lens, or generating data sets around what's the impact that can be generated with these different types of effects together with different types of organisms. And that's going to become more and more important as we start exploring large classes of compounds that are emerging, like antibiotics, pesticides, and other types of compounds.
If I can add also that my interest is also on trying to understand through genomics, like, because of this all mixed contaminants, and we can go for a specific contaminant if we want to, but we will never know the effect of all these things being present at the same time. And anything in the environment has to kind of cope with all of those. So whether genomics or metabolomics can be used to understand this cumulative effect, I think, which is missing. And this is quite essential that we understand it, because we may ourselves be getting exposed to many of the mixed situations where lots of contaminants of different types, whether it's in water, whether it's in air, or what we eat. So I think this is where I think the research has to go.
Dr. Wishart, final word to you.
Well, I think because it is time to wrap up, I really do want to thank our panelists today, David Lloyd and Shiv Prasher, for some really excellent comments and insights here. I also want to thank all of our attendees who've been able to stick with us right to the bitter end. This sort of last of the 4 presentations we've had on metabolomics, and I've certainly enjoyed the opportunity to reach out to the BioAlberta community and to others around Alberta and across Canada. I also wanted to thank our host, Rob Stoddard, and also Trish from the BioAlberta office for their help, and for Jen Reed, who's been here at every one of the presentations. So thank you, everyone. I really appreciate it.
Okay. Thank you.
Thanks, Dave. Thank you to David, and to Shiv for their time today. And on behalf of BioAlberta and our members, thanks to TMIC for partnering with us on this session. This has been a great opportunity to really go deep, broad and deep on a subject of interest. For those of you who attended this one, thank you for joining us today. If you have feedback on this session, please let us know, good, bad. or if you have information or ideas on different topics, reach out to us on social media and let us know. We would encourage you to sign up for BioAlberta's newsletter and events letter, so you can see what's coming in events or in this forum and our mentorship series and other ways to connect within our ecosystem. And I'd also encourage you to reach out to Dave and Jen at the at TMEC to give them their feedback, or if you have any more information. Thank you very much for attending today. Have a great evening, and we will see you soon. Take care.